I'm the team lead of Omega Bio-Edutainment. This morning, on our educational series, we have come to learn something on microbial genetics with focus. Microbial genetics with focus on two important topics. The first one is to actually consider to complete our lectures on gene regulation, gene expression, and gene regulation. So the aspect we are going to focus on today is going to be gene regulation. Then we will now look at introduction. We get to introduce ourselves to what is called recombinant DNA technology. So we are just going to be introductory part of recombinant DNA technology. However, let me try and run through what we have done before on gene expression. Don't forget that the first one is to we talk about how to understand how gene is being expressed, how is gene being expressed, especially in microorganisms. So the way eukaryotic organisms send out signals and use it to perform different type of tasks, it's the same way they express their gene. If gene, and don't forget that there is a very basic analogy. According to the law of evolution, especially the one that is related to Jean Lamarck, which is the theory of use and disuse. So there is no point in you as an organism, in you synthesizing the gene that you do not use often, genes that is not needed, genes that you do not have to make use of it often and often. Jean Lamarck actually said something about the theory of use and disuse. What he said was the trait or the organ that you tend to use more, your body, your system will develop it. It's just like learning a trade. The trade that you are making profits in it every time, you will always want to improve on it. But the other one that is just maybe occasional, so you don't need to actually improve on that. And that is the reason why gene expression is very, very important. And you should know that genes that are very vital, there are some genes that are vital. We call them essential genes. For example, in a pathogen, look at in a pathogen, the ability of a pathogen to continue to exist will depend on how virulent that pathogen is. That is, it may be able to develop certain genes that will make it to be more virulent, that will make it to be more to survive in the host very well, and we develop a mechanism that will make it be able to overcome or overpower, to overwhelm or overpower the immune mechanism of the host. So, and that's why I told you earlier on that the issue of host pathogen relationship is that the host will be looking for a way of winning the war. At the same time, the pathogen of looking at winning the war. So that is why we have to know how are these important genes, how are they synthesized? Why would they know when and when it actually needs? And that's why